Hi there, I hope you're doing well. My name is Edward Barry and this is my Biology 231 video project 2 on olfactory kin recognition between birds and their eggs. While the role of visual cues involved in the recognition of kin eggs has been well studied in birds, the role of olfaction has not been investigated much at all. This behavior may be important for having the ability to detect brood parasites, which reduce the host parent's direct fitness through killing the other offspring, or by taking resources from the direct offspring of the hosts. Having this ability to distinguish between kin and non-kin allows for parents to detect parasitism, both interspecific and conspecific. The first research into this topic was done by a Spanish team who used model eggs of the great spotted cuckoo to test a main species that these cuckoos parasitize, Eurasian magpies. In one experiment, they tested how the magpies responded to model eggs scented with tobacco smoke compared to an unscented control. They found that the magpies had statistically significant increases in ejection rates for the tobacco-scented model eggs compared to the unscented controls, constituting the first evidence that hosts of brood parasites use their olfactory ability to detect and eject foreign eggs from their nests. The study I am focusing on is titled Female Zebra Finches Smell Their Eggs and was authored by a German team. They used the model organism Taeniopygia guttata, or the zebra finch, to determine if zebra finches can discriminate between their own eggs and conspecific eggs based on olfaction alone. Zebra finches are a widely used model organism, in this case being very applicable because in lab populations have 21% of clutches in the nesting colony parasitized and 5.4% of offspring being brood parasites, which fits within the range for wild populations of this species, where 17.5 to 36% of clutches in a nesting colony are parasitized and 5.4% to 11% of offspring are parasites. For the testing that took place in this study, the researchers used a 3-compartment cage with a 115 by 40 by 30 centimeter main compartment, 15 by 15 by 15 centimeter nest boxes on either side, as seen inside the red circles inside the diagram. There were perches adjacent to each of these nest boxes. Each nest box with its adjacent perch was defined as a preference zone. These nest boxes contained coconut fibers to simulate a nest, and behind this was an egg, either an own egg or a conspecific egg, which was suspended in a cage with a fan behind it. The coconut fibers were made sure to obscure any possible visual cues of the egg. After all eggs were laid, testing was done on day 3 after clutch completion and on day 10 after clutch completion. The testing procedure for this experiment was as follows. A female was placed in the central compartment, with the nest boxes separated from the main compartment by opaque slides to block visual and olfactory cues. The fans were then turned on for 20 minutes to allow for the accumulation of egg odors inside the nest boxes. The opaque slides were then removed and the female's movements were recorded for 5 minutes. Her position was recorded every 3 seconds. The researchers used time in either preference zone, either that of the own eggs preference zone or of the conspecific eggs preference zone, to determine whether the zebra finches can determine their own eggs from conspecific eggs based on olfaction alone. If a bird went into neither preference zones, then their results were excluded. Researchers found that while the birds did not spend a statistically significant difference in time in either preference zone on day 3, and a statistically significant difference in their results from day 10. On both days, a sample size of 25 was achieved. We can see these results in the diagram here. In summary, the study provides the first evidence that birds can discriminate between their own egg and conspecific eggs based on olfactory cues alone. The study, at the very least in my opinion, shows that a zebra finch's preference for being around the smell of their own egg makes them go to their own egg, and at most can be used as part of evidence that birds use olfactory cues to recognize and possibly reject parasitic eggs. It is not yet known if the smell that the birds prefer is their own smell, which they transfer to their eggs from their preen glands secretions over time, or if this is the smell produced by the developing embryo inside the eggs. Both of these hypotheses have evidence behind them, and both could explain why the authors did not see a statistically significant difference on day 3, but they did on day 10. If this is solely due to preen gland secretions, then I think that detecting parasitic eggs under natural circumstances may not be possible. But if the developing embryo's odor is involved, then I believe that such discriminations could be possible. A further study was done in 2016 by a French team using Halobaena caerulea, or the blue petrel, as a study species in much the same fashion. There were some big methodological differences. For example, the researchers in this experiment used a Y maze, as pictured here, instead of a cage, and they did the study throughout days 9 to 21 after clutch completion, rather than on 2 separate days. Found a statistically significant difference in their results, with 82% or 14 out of the sample size of 17 of the birds preferring to approach their own egg in the Y maze over the conspecific eggs. They also found that the birds spent more of their time with their own eggs as opposed to the conspecific eggs, with an average of 5.7 minutes plus or minus a standard deviation of 0.9 minutes being spent with their own egg versus an average of 2.0 minutes plus or minus a standard deviation of 0.5 minutes being spent with the conspecific eggs, as is shown in the diagram. References, I hope you enjoyed my video. Thanks for watching.